Hey, good morning. I would like to welcome you all to celebrate Vision Research. It's Friday, 8:30, and that's what we're going to do today with our very special speaker. And fortunately, he's in person. Before, I think he gave this talk a few years ago, but that was Zoom. So we're really delighted to have you here. And before I say anything silly after that, I will ask Claire to introduce our speaker.
It's all okay. Good morning, everyone. My name is Claire Melly, and I'm a fourth-year medical student in Dr. Andrew Brown's lab here at UCI Center for Translational Research. It is my pleasure to introduce you to today's speaker, Dr. Richard Lang. Dr. Lang is a professor in the Department of Ophthalmology at the University of Cincinnati, as well as the Director of Visual Systems Group and co-director of the Science of Light Center at the Cincinnati Children's Hospital Research Foundation in the Division of Pediatric Ophthalmology. He received his PhD from the University of Melbourne at the Ludwig Institute for Cancer Research, followed by postdoctoral training at UCSF. He then went on as an assistant professor at NYU in the departments of cell biology and pathology in the School of Medicine, leading up to his current appointments at the University of Cincinnati. Dr. Lang has over 150 publications, many of which are in prestigious journals such as Nature, PNAS, and Cell Reports. He has been invited to speak in various seminars and teach all over the world, holding positions at Keio University in Tokyo, as well as Umeå University in Sweden. Dr. Lang has also received numerous honors and awards, such as the Research to Prevent Blindness Lou R. Wasserman Merit Award, and has mentored many PhD and postdoctoral fellows to successful careers. With that, please join me in welcoming Dr. Richard Lang in his talk titled Atypical Opsin Light Sensing in Development, Homeostasis, and Disease.
Thank you so much, Claire, for that introduction, and thanks to all of you for having me here today. As many of you will be aware, my lab has been very interested in lots of different aspects of eye development over the years. But in about the last decade, we've started focusing on light sensing pathways and their influence on development and homeostasis. I'd like to begin with this movie. This is, of course, a movie of planet Earth. It's from a geosynchronous satellite. And it's just a reminder about where we live and, more to the point, where we evolved. And so obviously, because this ball of rock in space rotates and there's a sun close by, we're washed daily with this pattern of EM radiation, and some of that comes to us in the form of photons we call light. And you'll also notice that this is focused on the most beautiful of the continents. But this has been, this maybe. Sorry, it's not advancing. What am I doing wrong? Any suggestions?
No, it says it is on. Tim, they said it's on.
Yeah, it's advancing.
Oh, there we go. Ah, right. Thank you. So the availability of these photons has been an opportunity for evolution to establish mechanisms that are adaptive. And of course, this audience knows very well that we have a visual system that allows us to decode the identity of objects based on the photons that bounce off of them. But you'll also be aware that we have things like light-entrained circadian clocks that establish a rhythmic physiology. Our physiology is fundamentally rhythmic, and these circadian clocks are part of the mechanism that establishes that. And my lab has been interested in a series of other questions as well about how light stimuli will influence eye development in particular, but it's also now known that light can regulate brain development, at least at some level. And I'll also touch on this idea that there are some light-dependent mechanisms for regulating energy homeostasis. So that's actually a pretty extensive biology. And that's why we're starting to use this phrase, sun-coupled physiology, because it's very clear at this point that our physiology is intimately coupled to the sunlight cycle. As you'll all be aware, we don't live in a natural lighting cycle. We live in circumstances like are shown in this large city in the bottom right, and then a close-up of a cityscape is a very different lighting circumstance, and we live with the ubiquitous electronic devices as well. And so there's this one concept that we can consider, and that is that we choose to live in opposition to our evolved physiology. And as I'm going to explain, that has consequences. One of the reasons I like working on opsins is I get to show the Palczewski structure, and that makes me happy. But as you're all aware, the opsins can absorb photons within a particular wavelength range. This chart just shows the selection of opsins you find in humans. My lab has been especially interested in the function of opium-3, opium-4, and opium-5. And as you notice on the chart, these tend to be the short wavelength opsins. And in particular, opium-5 has an absorption maximum around 380 nanometers. So this is this is the shortest wavelength absorption, and so that is of particular interest, as I'll explain. These molecules have been around for about 660 million years, but excepting for about the last 200 years, the major source of photon ligands for these molecules was the sun. So our interest in this biology really emerged from our interest in vascular development, and this schematic explains what we've discovered over the years about the way light-sensing pathways regulate vascular development in the mouse eye. This is a bit complicated, but basically what we showed was that the opium-4 melanopsin is actually involved in regulating the number of retinal neurons that arise during the developmental process. If this pathway fails, you get too many neurons. That has a consequence, because oxygen demand is increased, and then you induce VEGF-A, and that has an influence on the way the vasculature develops. On the other side of this schematic, we have opium-5 is actually involved in regulating the levels of dopamine within the eye during a postnatal period. And you may know that dopamine has an antivascular function because it can stimulate dopamine receptor 2 and actually influence the signaling activation of VEGF receptor 2. And so what this means is that these two light-sensing pathways have opposing activities. One of them promotes and one of them suppresses vascular development in the eye. And we look upon this as a timing feature of the way this development is controlled. One of the really interesting consequences of this work, one of the really interesting conclusions, was that since these pathways were functioning for opium-4 in late gestation in the mouse, and for opium-5 just after birth, they're actually functioning as deep tissue photosensors. So these are not, obviously, the eyelids are still closed during this phase, and this is late gestation in the mouse. So it was an indication that deep tissue photosensing could be relevant for mammalian development. We had this basic question, and that is how melanopsin might regulate retinal cellularity. And so the project I'm going to tell you about next is directed at that question. And this is Shane D'Souza. Shane is a super talented trainee who was first my graduate student and is now a postdoc in the lab. And everything I'll talk about in the next dozen slides is his work. So we took a very standard approach in the beginning, and we just identified the genes that were differentially regulated in an opium-4 null mouse. We mapped those differentially regulated genes onto the UMAP space for the single-cell sequencing data from Clark et al. We identified some patterns that are very distinctive, and they appeared to be within the rod photoreceptor lineage if you look at those cell type color codes. And we also use this technique called RNA-BEASC that allows you to estimate the proportion of cell types that are modified from bulk RNA-seq data. And what you can see in this chart in the bottom right is that it looks like rod photoreceptors are increased in number, and late retinal progenitors are reduced in number. Those are the two statistically significant changes that we identified. And so, of course, what this suggested was that maybe opin-4 and the intrinsically photosensitive retinal ganglion cells that express opin-4 might be regulating the number of rod photoreceptors. So that was a bit unusual, so we had to investigate that further. And obviously, when you're changing cell number in the retina, there are a couple of different ways it can happen. You can change the cell division rate, or you can change cell death. There was a question about whether cell death could occur in the rod lineage, and so we first simply investigated whether that was possible. And we took advantage of these two nice tools, NRL-Cree that actually came from Anand Swaroop's lab, and Sunone SFGFP that Jeremy Nathans generated. The NRL is, of course, as you know, a lineage-specific transcription factor, and this Cree restricts activity to the rod lineage. Sunone SFGFP is a nuclear envelope marker, and it's especially useful for counting cells that are very tightly packed. So we put those two together, and you could get, and also labeled with anti-activated caspase-3 antibodies, and that's just some of the histology showing there. But then we mapped all the events over a developmental time course onto a model of the retinal laminations. And you can see in green here where these events within the rod lineage arose. And there was plenty of cell death, first of all. And you could also show that they were located where you would expect rod photoreceptor precursors to be. And at P5 and P7, you could see that a significant proportion of the cell death that occurred in the retina at that time was within this lineage, particularly at P7. So we knew we had to focus our analysis efforts. And on the right-hand side here, we've actually matched our quantification of rod lineage cell death to that was produced a long time ago by Young. And you can see that it's quantitatively identical. So this was just a very nice confirmation that he was correct. His was simply an anatomical assessment of whether cell death was occurring in rod photoreceptors. And so it's very clear that that was accurate. And so obviously we've just shown that programmed cell death can occur within the rod lineage. So then we did the obvious experiments and asked whether we could modify the amount of cell death that occurred in rod photoreceptor lineage with an opin-4 loss of function. And so you can see here in the blue bars here that the opin-4 null animals have a reduced level of cell death in a nuclear layer, which is where those cells reside. And we could not find any evidence for a change in the proliferation rate within this lineage. And then the crucial experiment was probably this one, where we used the lineage mark that I've already described and did a dark reading experiment and simply showed that you could reduce the rate of programmed cell death and quantified here, showing you that the dark read condition dramatically reduces the number of rod lineage cell death events that occur. So in other words, this is pretty good evidence that both melanopsin and light actually regulate the number of rod photoreceptors that arise. And you can demonstrate that indeed you do change the number of rod photoreceptors if you quantify them later during development. You can see this band of NRL positive cells in the inner nuclear layer in the opin-4 null that doesn't exist in the wild type. And this is the increase in the number of NRL rods that you can observe. And then also in the dark reading experiment, you can see this expanded domain and an increase in the number of rods. There is no consequence for cones as a result of these manipulations as far as we can tell. So obviously this is consistent with a model in which rod numbers are regulated developmentally by light and an opin-4 pathway. So you will realize that we have a little bit of an issue here because the IPIGCs, being in the retinal ganglion cell layer, and rod precursors are well separated in the laminations of the retina. And so we considered various mechanisms by which the IPIGCs could communicate with the rod progenitors. Some of those ideas were poor ideas, but we realized in the literature that there was a very interesting observation that others had made. Sam Hattar's group and David Burson's group had identified something that they called an outer retinal dendrite that was associated with melanopsin expressing retinal ganglion cells. And you can see them here. So this is just a visualization of processes from an OPN4 IL-14 mouse, and you can see all these processes extending up into the retinal layers, and these are, of course, IPIDC processes. We use the more sparse labeling reporter to do a bit more of a detailed analysis, and you can see here, if you look in the Z-plane, these processes that are extending up into the outer retina. And so these are also transient. You find plenty of them at the day of birth, but then they diminish in number pretty rapidly after that. So consistent with the idea that you're getting this light stimulation in this postnatal period. And they're unique to OPN4 expressing retinal ganglion cells. If you look at the OPN5 lineage, we have all the tools for doing that. You don't find any of these processes. And so we wondered whether these were the way in which the IPIDCs communicate the light information to the outer retina where the rod precursors reside. And so we can basically demonstrate that quite nicely by doing a light stimulation and showing that you could get a FOS induction. This is the dark control, and this is a 470 nanometer stimulation. And indeed, if you look closely, you can identify FOS induction in IPIDCs that have these processes extending into the outer retina. And so that seemed to be consistent with this idea. Okay, so we looked more closely at the features of these processes. And one of the things we could show was that the photopigment melanopsin is actually in the processes. And you can see that here, these couple of examples. We were very curious about the possibility that glutamate might be involved in this, because one of the things that IPIDCs do is signal with glutamate. And this labeling shows that you can actually see a component of the glutamate packaging machinery, VGLUT2, in the processes in the outer retina. And so it seemed like maybe they could even function locally in sensing light and eliciting responses in the outer retina. And I won't show you the data for this, but curiously enough, these processes also have synaptophysin in them. And synaptophysin is usually associated with a presynaptic structure. And so the idea that these are a dendrite is accurate anatomically, but they have these some molecular characteristics of axons, it seems. And so we view them as some kind of a strange hybrid neurite. And that we haven't really got to the bottom of their exact nature yet. But obviously this suggested that maybe these processes could release glutamate in response to a light stimulation. So we did the obvious experiment and actually did a conditional deletion of SLC17A6, the gene that encodes VGLUT2, with OPN4 Cree. So obviously an IPIDC specific deletion of this glutamate packaging machinery. And sure enough, you could see diminished levels of programmed cell death in the outer retina. And that chart just shows spatially that the diminished level of cell death occurs in the right part of the retina to be consistent with rod photoreceptor precursors not dying. So obviously one requirement of this model where we're suggesting that IPIDCs could deliver glutamate to rod photoreceptor precursors is the existence of a glutamate receptor in the rod photoreceptor precursors. And so we went back to all the single cell sequencing data that's out there and reanalyzed it for glutamate receptors that might be good candidates. And so this is the rod lineage represented in the single cell sequencing data, and this is a pseudotime axis. And if you plot the expression of the glutamate receptors, there are many of them, of course, onto the pseudotime axis, you find that one of them, GRIC3, has this really provocative expression pattern, which is transient and quite consistent with the phase of activity of this light response. And so for comparison, this is NRL, the transcription factor that we know is functional in the rod lineage. And Anand Swaroop's group had also done some experiments where they flow sorted NRL EGFP cells. And looking at those data, we could show that within those data as well, there was this transient expression of GRIP3 that was consistent with the pseudotime axis. So this seemed like a really good candidate. And so we could also show with immunofluorescence that you could identify GRIP3. This is just antibody labeling. You can identify GRIP3 in that part of the INL where we'd expect the action to be taking place. So our first attempt to do the functional study was actually to generate a GRIC3 flocked allele. And of course, the goal was to do a conditional deletion within our CRE. And for reasons we don't quite understand yet, we cannot make any homozygous conditional deletion mice. Some quirk of the genetics is defeating us there. However, we took up an alternative, which is to do a somatic deletion of GRIC3 using an electroporation strategy. And the fact that this experiment happened at all, this is a really difficult experiment, the fact that it happened at all is a good reflection on Shane D'Souza's talents. But basically what we did was co-electroporate these two plasmids, one of which is NRL DS red, and this simply marks the rod lineage, of course. And then we used two different plasmids that had different guide RNAs, one of which was a control and one of which was targeted GRIC3. The electroporation had to be done on the day of birth because of the timing, and that was perhaps the trickiest part. And of course, because of the combination of plasmids here, and because we're also labeling for anti-activated caspase 3, you get this sort of color-coded cell pattern here. And you can decode that information. You can map the different colors onto the retina, and it maps nicely. You can show that cells of a particular type are where they should be. But the important result is this one, where you can show that when you actually target GRIC3 with the guide RNA that should inactivate GRIC3, you can get a dramatically reduced level of programmed cell death in rod lineage cells. And so this is obviously suggesting that GRIC3 is part of this pathway as well. We hope to solve a little genetic problem with the conditional allele, but this is at least some data that GRIC3 is involved. And so the conclusion from this part is that we appear to have a mechanism by which melanopsin in iPRGCs communicates with these rod precursors in the outer retina through glutamate and GRIC3 to actually regulate the number of rod photoreceptors that arise. And we think about this as an adaptive developmental process. And so this will eventually get there, I think. We think about this as a possible mechanism for fine-tuning the function of the retina for different environments. And so, for example, if you have this kind of highlight environment, maybe it's advantageous to actually diminish the number of rod photoreceptors you have, perhaps in favor of other cell types. Or if you are an animal that lives in the understory in a low light environment, maybe you want to keep all those rod photoreceptors intact. And so, this is just one way to think about this. We have done one experiment with Petri Ala-Lorela, who you may all know has this wonderful assay for single photon responses in rods. We sent him some mice and he assessed them for us, and we could not detect a difference. And so that says that this is not simply about sensitivity. And so we think there's probably going to be some other feature of retinal function that is modified by this pathway, and we're going to have to look much more carefully at the circuitry. But this in general is the way we think about this. So now I think, now we've sort of got the developmental mechanism down, we can think much more carefully about what this means functionally and behaviorally. Okay, so coming back to our origins in light sensing pathways, one of the conclusions we drew was that opin-5 was actually involved in regulating levels of dopamine within the eye. And you'll all be aware that dopamine has been implicated in the etiology of myopia. So when we learned this, we got pretty excited about the prospects, because the function of opin-5 in a myopia pathway might be consistent with all the data. So let me explain. So obviously you know what myopia is. An axial myopia is where the eye grows too long and the images are focused in front of the retina. It's a huge public health problem. These are data from the WHO showing what's happening with myopia. And so today there are about 2.8 billion people with myopia. And by 2050, we're going to have about 5 billion people with myopia. And so this is by far the most common disease. And there's actually a very nice article from Ellie Dolgen that describes this as the myopia boom, and it sort of discusses all the issues. And as I understand it, Ellie is producing an update on all of that we hope to see fairly soon. And so that's a really good article to read to get up to date with all of this. So obviously we have this big question, what is causing the myopia boom? We know that establishing perfect focus in the eye is dependent on light stimuli. That's been demonstrated by many different groups over many years. And there's a process called emmetropization that we use to describe the establishment of perfect focus. It's been the question of whether this has a genetic cause has been considered. And there are a whole series of GWAS studies that have been performed by the 23andMe and CREAM consortia. They found a whole lot of risk-associated genes that made sense. They were things that function in the retina and so on, in various parts of the eye. The problem with those studies was the effect sizes were small. And so it suggested that we could not explain this with a genetic cause. And also, it's just the progression here is just much too fast. How are you going to propagate risk alleles through a whole population like this over this period of time? So the genetic cause is not a good explanation. And so we end up with a situation where we're trying to explain this as a developmental origin problem. So there's a vast literature of myopia epidemiology that some of you may be familiar with. And there are a couple of main conclusions from that literature. One is that sunlight and outside time is protective. The second is that inside time is a risk factor. And so all of that is consistent with the idea that there's an environmental origin of this. When we were doing the work on opium 5 and regulation of vascular development, we were aware that our colleagues in Japan, Toshiko Uehara and Kazuo Tsuboku, were very interested in the possibility that violet light was involved in the regulation of myopia. And so putting all of those things together, it rather suggested to us that opium 5 was going to be involved in the etiology of myopia because it's a violet-sensitive opsin, of course. It regulates dopamine. It's been implicated in this disease. And so we decided to pursue this question more carefully. And so this is just rolling through very slowly. So we produced, as a result of a wonderful collaboration with both Michel Pardieu, who you may know pioneered the use of the mouse in myopia studies, and Toshiko Uehara and Kazuo Tsuboku, we produced this paper that had some very interesting conclusions. We got in touch with Toshi and Kazuo because we knew they had a very nice model for doing myopia in the mouse. And so this is a mouse fitted with spectacles. And the way you do this is that one of those spectacles has a -30 diopter lens. And so that focuses images behind the retina. The response of the eye is to grow long. So this is an induced axial myopia. And then on the other side, you have a 0 diopter lens, and that's your control. And basically, most of this analysis is done by comparing the two eyes. And so, of course, you image the eyes with OCT, and you do you assess the refraction with an autorefractor, and you can get data very nicely that way. The main conclusions of this study were as follows. The first was that 380 nanometer violet light at the opium 5 lambda max could suppress and induce myopia. And I'll show you some of the data for that. And then the second conclusion was that violet light suppression of myopia was dependent on opium 5. And so we also learned that the time of violet light delivery was crucial. In these experiments, we actually delivered light at dusk. And that matters, we think. And so these are some of the data that I think is just worth emphasizing. So when you do these experiments, this is refraction. So when you do these experiments, the eye with a -30 diopter lens shifts its refraction down. And in a mouse, you get it to approximately -30 diopters, makes sense. And so this is the shift you see in the opium 5 mutant mouse. So this is a Rex-free opium 5 flox conditional mutant. So you're only targeting retinal neurons. And you can see it shifts just fine if you use standard white LED, the kind of light you have in this room. By contrast, if you do that same experiment, but you do it in standard white light from an LED, but you add in 380 nanometer light in the evening, you'll notice that the opium 5 null still shifts. It doesn't change its response, but the wild type mouse doesn't shift. And so this is a pretty good indication that as far as refraction is concerned, you can actually prevent the induced shift with violet light. And it's opium 5 dependent. And on the right-hand side here, you just see the axial length version of those same experiments. So you can see the axial length increases for both genotypes in standard white light. But when you add in the 380 in the evening, it's the wild type that doesn't elongate. And so this is pretty provocative because what it says, and so it's kind of a remarkable result too, because you've got to remember that these eyes that are not shifting in the presence of violet light still have the -30 diopter lens in front of them. So this is not about refraction. It's about something else, something much more fundamental. And so obviously this is consistent with the idea that opium-5 and violet light protect against a myopogenic stimulus. And of course, this is a very aggressive myopogenic stimulus. The other data that is worth looking at, because it's really interesting in the same way, is the choroidal thickness data. So this was not actually published in this form. This is the raw data for choroidal thickness. And normally when you induce a myopic shift, you see choroidal thinning. You can see that here. And no opium-5 mice here, just wild-type mice. And you see with the 0 diopter lens, this choroidal thickness increase is just part of normal development over the 3-week period. But what you notice, if you add violet light in the evening, everything looks normal. You prevent this choroidal thinning in the -30 diopter lens. So again, it's the same thing. You can prevent the changes associated with myopia with violet light, even with the -30 diopter lens in place. Okay, so that's the conclusion. And so if you look at these data, plus other data we have about the function of atypical opsins in refractive development of myopia, this is a good conclusion. So recently we showed that the opium-3 germline null mouse has a refractive development issue. We don't know much more than that right now. And Michel Pardew's group, this Chakraborty paper, and Liu et al. In China have also shown that opium-4 null mice have a problem with refractive development and induced myopia. So in other words, all of the short wavelength atypical opsins are somehow involved in this process. And I mentioned time of day stimuli. Certainly, it's been shown that opium-5 is associated with circadian clocks. I'll come back to that. And it's been shown that time of day light stimuli probably matter for this biology. Okay, so how does this explain the myopia boom? Well, we think it looks like this. So this is really a causation argument for the myopia boom. We spend 93% of our time inside in environments like this one. There are no violet photons in this space. And so if you look on the right-hand side here, this illustrates the problem. This is a representation of sunlight, and of course you can see it going down to 350 nanometers here. This is a fluorescent light, and fluorescent lights are built with they make short wavelength photons that actually stimulate a bunch of phosphors that are sprayed on the inside of the tube, so they produce all these peaks. That's what you see here. And then a standard white LED, and this is the most common form of lighting these days. This was a Nobel Prize winning invention, and it's a Nobel Prize winning invention because it's incredibly energy efficient, and it's going to help solve our energy consumption problems. And typically they have a peak at 450 nanometers, and then there are phosphors incorporated into the design to give you these long wavelengths. And this is fantastic for vision, but of course totally misses the stimuli for the short wavelength photons. And so there's also a problem with modern glazing. Modern glazing is designed to be energy efficient, so it excludes a lot of the short wavelengths and actually a lot of the long wavelengths as well. So you end up with environments that are deficient in those wavelengths. The digital display devices are RGB devices that look a lot like this in their output, and they, again, do not produce these wavelengths. So if we go back to our chart that describes the absorption spectra for the different opsins, in the inside space, you're really talking about something that looks like this, where these short wavelengths are missing. And so we think that this is probably our problem, that if these short wavelengths are involved in suppressing myopia, then their absence in the belt space, where we spend most of our time, is going to mean that if you are subject to a myopogenic stimulus, you're not able to correct. Okay, so the mouse experiments were really powerful, but we felt that it was really important to go beyond the mouse and do some experiments in something that was nearly a primate, the tree shrew. So you probably all know this gentleman, Ruffy Gritz. So Ruffy is at UAB, and he's trained as a bioengineer. He has a very strong interest in myopia and a number of other visual system diseases. Ruffy also has the largest tree shrew colony in North America, and he's done some fantastic work with this. So a long time back, Michel Pardue kept telling me, you have to talk to Ruffy, you have to talk to Ruffy. So I spoke to Ruffy, and we started this collaboration that I'll describe now. And so it's been wonderful working with Ruffy because he's an engineer, and this is a very device-intensive project. It's great because when we need a device, he just builds it. It's very simple. And so this is the northern tree shrew, Tupaia belangeri. It's a relatively small animal, and it's been developed as a wonderful myopia model. It was actually Tom Dawkins who developed this as a myopia model some time back, and the people at UAB have been using this for a while. They show this extremely reliable myopia response. So I actually don't have to use many animals compared with a mouse to do a sensible experiment, as I'll show you. But the really big advantage of the tree shrew, besides all of that, is that it's nearly a primate. So it used to be classified as a primate, but if you look here, it now has its own order called a Scandentia, but it's immediately adjacent to the primates. And obviously the goal here is that if we're going to learn something useful in the tree shrew, it's probably going to be precisely relevant to the human. So those are all reasons to use the tree shrew for this work. Now, one of the things we had to deal with in starting this work was that we were already aware that the human lens does not transmit 380 nanometer photons very effectively. That's been documented multiple times. This chart actually has drawn on data from the literature. You can see the sources listed here. And these colored curves show the lens transmission to particular wavelengths. And so you can see it's age dependent. So early on, so this is a 4.5-year-old lens, and you can see there's a little bit of transmission between 300 and 350, and kind of a decent shoulder here. And this is the absorption spectrum of human opium-5. And so you can see there isn't a lot of matchup. So if you want to think about stimulating opium-5 in a human retina, you really have to think about maybe this early age, or maybe this shoulder region here, okay? And so Ruffy did some calculations and showed that if you wanted to stimulate opium-5 in the human retina during the period of time where there's myopia susceptibility, you are going to have to do that using photons that are somewhere between, what is that, 413 and 416 nanometers. That seems to be the optimal. This is where Ruffy's engineering skills come in, because of course we wanted to have that data from the tree shrew as well. And so Ruffy built a device for measuring the wavelength transmission through tree shrew lenses. You can dissect them out very nicely. You can put them in the pathway of this optical device, and you can use a spectrometer to measure what the transmission is. And so this chart that will come up in just a second is the equivalent of the human data I just showed you. And so again, these colored curves show there's a stage-dependent transmission characteristic for the tree shrew lens. This young animal here appears to have some transmission within the opium-5 stimulation peak. But as they get older, of course, you are shifting to the right. So again, you're talking about maybe early stimulation, but the shoulder region stimulation. And if you just take all that information into account, Ruffy again did that calculation for optimal stimulation and argued it would be between 420 and 429 nanometers. So then we did, and so the conclusion here is that if you really accept the idea that opium-5 is involved in suppression of myopia, the optimal wavelength for suppressing myopia in humans may be different from that in the mouse. So we tested that. And this is just the summary showing you the optimal wavelengths for stimulating opium-5 in those circumstances. So then we did a whole series of experiments in the tree shrew with induced myopia. And of course, this is a -10 diopter lens in one eye. And we supplemented the ambient lighting with with these violet indigo wavelengths in the range I've described. And these are some of the data. So these are the refraction data. So Ruffy also built these ambient light arrays. So he took a standard white LED as the main illumination, and then he supplemented with a LED that was either, that was in this violet to indigo range. And so this chart's a little bit complicated, but so this is normal. This green line is normal. And this is just refraction. This black line is the induced myopia. And so this is what happens at a -10 stimulus, you get to -10, and that's the perfect matchup in the tree shrew. And those two are in the presence of the standard white LED. If you start adding in the violet and indigo wavelengths, and what we did was add them at the beginning of the day, at dawn, and we did that because obviously this is a diurnal species. And we imagined that maybe compared with the mouse, it was going to be dawn that mattered. So we added those indigo and violet wavelengths at the beginning of the day, and you can see that wavelength really matters. And so here is 365 and 395 nanometers down here. So they really don't have much impact at all. When you get to 400 nanometers, you are suppressing the change in refraction a little bit. But when you go to 420 nanometers, it is a complete suppression, 100% or close to it. The axial length data are very similar in that you've got, I think the 365 and the 395 nanometers are up in here. In other words, you don't really make much change in the induced axial elongation. 400 nanometers has some effect, and here's 420 nanometers. You can completely suppress the elongation, and it's either a little bit less than the control. And so, in essence, it does appear, so 420 nanometer light is the most effective, delivered at dawn is effective. And what this tells us is that suppression of myopia in this species is exquisitely wavelength sensitive. The distinction between success and failure is 25 nanometers. And so another feature of these responses that's really important is they have almost no impact on the control eye. And so this is refraction in the control eyes, and you can see basically nothing changes. There is actually a statistical significance there. And then for axial elongation, you see axial length, you see something of a change, but minor, and probably functionally not crucial because the refraction appears to be normal. And so this is really interesting because what it's telling us is that these short wavelength photons are only changing the way the eye behaves in the presence of a myopogenic stimulus. So it's kind of a perfect situation. And this is the conclusion. This is just a summary for these different wavelengths. And you can see that just because of the LEDs we could buy, the amount of 420 nanometer light we used was a lot less than the other wavelengths I talked about. But here's the efficacy. You can get to 100% suppression of an induced myopia with 420 nanometer light. And we're pretty excited about this because we think this can explain the myopia burden. And so obviously we'll come back to this causation argument. We've made an argument that our inside spaces, where we spend practically all our time, are deficient in these photons. And obviously we're arguing that if you are subject to, so in other words, your ideal lighting system might look more like this, where you have that 420 nanometer peak. You probably have to make it circadian as well, so it goes through a rhythm every day, because that probably matters. But we're arguing that if you change the lighting environment, you can probably prevent this disease. That's the big question. And so while we're taking that idea to heart, and we're actually doing a pilot clinical trial now at Children's Hospital that is addressing this question. We have we're working with a lighting company called Bios Lighting, and they make these desk lamps that are circadian desk lamps, and we've persuaded them to put the crucial wavelengths in their design, and they've done that for us. And so we're going to send a bunch of kids home with this lamp and ask them to use them in the morning and in the evening as they play or study. And so the goal is to ask whether, and these are kids who've been pulled into the study because they have the beginnings of a myopic progression. So we're going to ask whether we can influence that. And so some of my colleagues in the architecture world are starting to take notice of this because, of course, the design of our buildings is probably the problem. And so these two, Lisa Heschong and Kevin Hauser, are research architects who I've discussed this problem with, and we published this paper that talks about this problem and how we can address it. And then mostly for entertainment's sake, I want to tell you about a conversation I had with Apple computers. And so back in the beginning, when we first discovered that violet indigo wavelengths were going to be important for suppression of myopia, we realized that if you could change display devices so they had better characteristics, you could probably very effectively deliver the short wavelengths and prevent myopia. So we talked to them about this. They have a vision scientist, and we talked to the vision scientist about all of this, and they listened very politely and went away and didn't contact us again. But then they produced a year later this app, and the app says, go outside every day, don't stay inside all the time, and hold your iPhone like this, far away from your face. And so, it turns out that the risk of liability can be a motivation. So, things are starting to move on this front. Okay, so in the last little while, I'm going to take you outside the eye and tell you about some of the other biological pathways we're working on. And I thought I'd start with this. This is just a schematic that shows you all the light-responsive tissues in a mouse. So you should be aware that my colleagues in the light response pathway world have basically denied the idea that mammals have extraocular light sensing. They don't like this idea at all. But the idea that mammals don't have this is clearly wrong based on everything we've found. So I thought I'd take you through some of the summary data. So obviously we know we have an eye and a retina that is light sensing. But in work that I'll summarize for you briefly, we know that there are preoptic area neurons in the hypothalamus that are directly light sensing in a mouse. They function as a deep brain photoreceptor. They've got very good evidence for that. I should add that in that, we've taken very careful measurements of photon flux deep within the brain, and there's sufficient photons to elicit a response from an opsin like opin-5. We also have melanocyte precursors in the mouse generally, but especially in the nose and in the ears that express opin-5 and actually regulate the function of a circadian clock in the skin. We have white adipose tissue that is light responsive, as I'll explain in a bit more detail. The trigeminal ganglion is light sensing, and as are the dorsal root ganglia. And so obviously, this is especially interesting for the trigeminal and the dorsal root ganglia because this is the major sensory input for a mammal. And so, we've done some basic experiments to show that they can sense light, and this is just the movie showing that they express opin-5 and opin-3. And so we know that opin-5 light sensing happens in TRGs. So obviously this means that a mammal is probably constantly sensing light within the environment and sending that information back to the brain, perhaps using it locally for whatever physiology is relevant. But we don't know very much more than that right now, but obviously we're addressing those questions. So obviously this suggests that mammals, like many other species, are set up systemically to sense light as part of normal physiology. And this is one of the reasons why I like that phrase, sun-coupled physiology. So intimate linkage to the sunlight cycle is where we are. So just a word about circadian clocks. Obviously, you know that there is one pathway that goes from the eye to the suprachiasmatic nucleus, where we have a so-called central circadian clock. That is a very well characterized pathway. Many of my colleagues in the circadian world have worked on that. But what has also been shown by a number of investigators, Tassini and Menneker, and more recently Ethan Buer, is that the retina has its own circadian clock, and it is dependent on light stimulation through opin-5 for entrainment. And you can demonstrate this very nicely. If you disable this pathway genetically, you can show that the retina continues to entrain to the light-dark cycle. And so it is clearly independent of this circadian clock. And then Ethan, with a little bit of help from us, also showed that those opin-5 expressing cells in the skin mediate entrainment of a skin circadian clock that functions independently of this pathway and this pathway. And so obviously this says that we have at least one extraocular photoentrainable circadian clock, and given all these other domains of opsin expression, it raises a very interesting question about exactly how many we have. And so we have to think about that much more carefully. Now, I should add that circadian clock responses is just one way you can demonstrate the function of extraocular light sensors. A lot of the light sensing responses are acute. You don't have to have a circadian clock machinery behind those pathways to make them work. You can elicit acute responses as well. Okay, so coming back to this hypothalamic domain of opin-5 expression. So this region of the preoptic area is crucial for homeostatic control of energy metabolism, including body temperature, as I'll show you. But it also it can also regulate food consumption, for example. The other domain I mentioned is expression of opin-3 within white adipocytes. And of course, these are just subcutaneous, many of them, and so they're going to be easy to illuminate with photons. And we've understood how these pathways, how these tissues are involved in light sensing. And so this is the summary for the adipose tissue work. We've published this now and shown that a blue-violet stimulation of white adipocytes actually enhances the lipolysis response. So this is the pathway that gives you the production of free fatty acids that are used as heating and energy fuel throughout the body. And this, for example, explains why the opin-3 null mice have a body temperature phenotype, because a lot of that fuel goes to the brown adipose tissue for heating. This is actually data from another group. The PI of the group his name is Peter Light. So you can't make that up. But what Peter's group showed was that human and mouse adipocytes both changed their electrophysiological properties in response to a blue light stimulation. And so here's the human response, and here's the mouse response. And so coupled with our work, I mean, Peter didn't know he was working on opin-3 at this point, but that's probably what that's probably the explanation for this. Putting all of that together, it's pretty clear that this is a conserved response and that it's real. And so let me show you some of the phenotypes of the opin-3 null because they're kind of interesting. So this is a cold exposure assay for opin-3 germline null and control mouse. And so you put the animal at 4 degrees and you just monitor body temperature. This is a rectal temperature. And you can show, obviously, the mice defend their body temperature. They upregulate their brown adipose tissue activity. And this is the way that a wild-type mouse behaves. This is the opin-3 null. It has a low body temperature. The explanation for that is that the lipolysis pathway is not fully functional. And then if you take that exact same cohort of mice, and instead of doing it in full spectrum light, you take away the blue photons, you actually show that the wild-type mice drop their body temperature to the same abnormally low body temperature as the opin-3 null mice. And so this is suggesting this is a blue light dependent, opin-3 dependent elevation in body temperature normally. And so this is actually a developmental study. If you raise animals in the absence of blue light, you can get kind of a mild version of that same opin-3 null phenotype. So there's a developmental component to this as well. And then you can do experiments like this one, where we've actually deleted opin-3 conditionally in white adipocytes using this adipo-Q-Cre activity. And you can show that those animals also have a reduced ability to defend their body temperature. And in this experiment, this is a 5-hour experiment. If you turn off the blue light halfway through, you show this descent in the body temperature of the wild-type mice to match the opin-3 conditional null. So this says that there's an opin-3 dependent, blue light dependent, adipocyte dependent elevation in body temperature normally. Okay, so coming back to the hypothalamus expression of opin-5, the preoptic area, we did some experiments to trace from those neurons, and we did it in a couple of different ways. We basically went out and we came back in, and it was very clear when we did all of that that this was going to be a thermogenesis circuit based on lots of work that others have done. And so we started to assess the possibility of a body temperature phenotype in these animals, and there was a pretty dramatic one. So an opin-5 germline null animals have a constitutively high body temperature. This is their body temperature response in a cold stimulation experiment. Here's the wild types. If you look at the brown adipose tissue, you find that thermogenesis genes are dramatically upregulated in this animal. And using thermography, these two animals on the right are the opin-5 nulls, Also, they look hot compared with the control. Temperature telemetry sensors show that the opium 5 null, shown in the blue trace here, still has a circadian pattern of body temperature regulation, but it's just constitutively high compared with the control, and the delta is the largest during the light phase, which might make sense. And then, not surprisingly, these animals have elevated energy expenditure according to indirect calorimetry experiments. One of the experiments we did was just ask whether the absence of violet photons could be equivalent to the absence of opium 5. And so in this experiment, we simply compared the opium 5 null phenotype with the phenotype of animals that are raised in the absence of violet light. And basically what you get is a mild version of the opium 5 phenotype. And so I showed you elevated expression of thermogenesis genes in the opium 5 null, and there is one significant change, UCP1, in the animals that are raised in the absence of violet. One feature of the opium 5 null is that the sympathetic nervous system innervation of the brown adipose tissue is upregulated. You see many more Th positive puncta in that brown adipose tissue, and you can quantify that. And you can also show that in animals raised in the absence of violet light, you get a very similar change. And so, and then the cold exposure assay, the kind I've already shown you, you can see this mild version of the opium 5 phenotype in those animals. So this is kind of remarkable because it says that just taking away violet photons can give you kind of a subtle but fundamentally different physiology, a fundamentally different energy physiology. And of course, we're thinking about this in the same terms we think about the myopia problem. Is this an explanation for the dramatically increasing incidence of the metabolic deregulation diseases? So we did one experiment to show that in a slice culture, a brain slice culture, the opium 5 neurons of the hypothalamus are directly light sensing. And so we used a FRET sensor for cyclic AMP to demonstrate this. This is an opium 5 null exposed to violet light, no response. The wild type, you get this nice response. They both respond to phospho and IBMX, the positive control. And so these cells are clearly directly light sensing. And this is the summary. And so if you put all that together, you kind of get to this place where we're suggesting that opium 5 in the hypothalamus is a deep brain photosensor, and that violet light is actually suppressing the activity of brown adipose tissue through the sympathetic nervous system. And then we're arguing that there is a blue light stimulation option in white adipocytes that results in the production of free fatty acids. And of course, we're arguing that this is going to be part of an energy homeostasis circuit because there's the possibility of sensory feedback. Heat itself may be feedback because these neurons are heat sensitive. And actually in recent work, we've been assessing the activity of endocrine mediators, and it's clear that leptin is involved in the circuit to some degree. But we're arguing that this is involved in regulating a lot of different energy homeostasis features, including the body temperature cycle. And I will, obviously the conclusion here is that we have an entirely new mechanism for regulating energy homeostasis that's in which we should really be coupled to the sunlight cycle. So I just wanted to show you this. Okay, this doesn't seem like the animation is working here, but I can explain anyway. So this is a mouse is a nocturnal animal. And so of course, during the inactive phase, which is daytime, its body temperature is low and it's higher at night when it's active. And we think what we've discovered explains this quite nicely because during the day, the inactive phase, you have light stimulating opium 5, suppressing the activity of BAT. And so you end up with a low body temperature. And at the same time, you're getting light stimulation of opium 3 in the white adipocytes, giving you free fatty acids. And that probably makes sense because when it's sleeping, the mouse isn't eating and you need an alternative energy source. Then when you go to the night, maybe this, here we go. So this is just the text explanation of that. So when you go to the night, you of course don't have any of this light stimuli. You're not suppressing bad activity, so body temperature can go up. And you don't need to stimulate opium 3 and enhance the lipolysis pathway because the mouse is eating and it has a food energy source. And so we think that works quite nicely to explain how body temperature variation occurs. And of course, we're thinking about whether these pathways can explain other aspects of energy homeostasis as well. And so this is the final summary slide. Obviously, what I've told you is that these light sensing pathways for opium 3, opium 4, and opium 5 are responsible for developmental and homeostatic functions that probably have quite important implications for human disease. And so obviously our ongoing work is going to continue to address all of these questions, and particularly try and couple this biology to the human disease questions more precisely. So this is my crew. This is a relatively recent picture of my crew. And the names in red are folks who've contributed directly to the work I've described today. So Gary Knight, Kevin Zhang, Brian Upton, Courtney Linney, Shruti Vembaraju, and Shane D'Souza. And I've had a set of wonderful collaborators over many years now. And as you'll realize, the way the lab works, none of this would have happened without all of these collaborators, and particularly Mike Yvonne, Ethan Buren, Russ Van Gelder, Randy Seeley, who's in Michigan, who's a metabolism expert. I mentioned Michelle Padu, Toshi Kurihara, Kazuo Tsubota, Rafi Gritz, of course, and then Takahisa Nakamura and Juan Sanchez Gamaches at my place. So with that, I will stop and I will be happy to take any questions.
Thank you very much. We have many questions, so we will try to speed it up and give a chance to everybody to ask. Give me a chance. My first question, a very simple one. In your experiments with children with different light displays, do those children wear glasses?
Some of them do. And
so that beats the purpose, right?
No, because we've changed the glasses.
So now they have a glass?
Now
plastic?
they have something called CR39 that allows transmission. Yeah, we've taken that into account.
Thank you very much. That was a good question and answer. Let's go to Osim. Osim, are you there? Akarash, are you there?
Are you able to hear me?
Yes.
Yes, I can hear you.
Oh, sorry. I thought my mic was not working. So I have a couple questions. I will ask one for now, if there's time, maybe more. So relating to the OPN4 story with the retinal vasculature.
Yes.
So in the studies showing OPN4 influencing the immature rod apoptosis, did you evaluate the number of brain3V ganglion cells and calretin and amacrine cells in regards to the 2013 Rao paper? Can you speak on how the findings from the glutamate signaling for rods may apply to what you found in 2013?
Yeah, we really haven't gone back to assess those cell types, but you're right in thinking that they may also be modified. The transcriptome analysis we didn't pick up signatures for those cell types this time around, but I do think it's an important question we have to go back to. So basically, I had no new information on that, unfortunately.
Thank you.
All right, let's go to Cher.
Hi, I just have a little question here. So if I understand correctly, children, at least human children, when they're born, they're mildly hyperopic. So if your mechanism requires myopic stimulus, does it mean that it prevents progression rather than stops induction?
Well, I'm not sure I know exactly how to think about this right now, because we have no idea about the mechanism underlying the response. But I would imagine, based on everything we know, I would think we have a pretty early stage violet influence on the human retina. But as I say, we don't know the underlying mechanism, so it's difficult for me to be precise.
Okay, thank you. Thanks, Chris.
Osim, are you there?
Yes, I'm here.
All right.
Okay, so my next question was, do you believe IPRGCs impact the regressing hyaloid and retinal vasculature in a direct, indirect, or possibly both mechanism? So far in your studies, you're suggesting that the main way melanopsin regulates the vasculature is by regulating the developing retinal cellularity. In any of these studies, have you seen a way in which IPRGCs may directly communicate with the retinal vasculature? So we know conventional retinal ganglion cells do, they influence the astrocytes, and recently y'all have shown ganglion cells release dopamine in early postnatal development. Yeah, no, it's a great question. I wouldn't exclude other mechanisms of involvement. I mean, the 2013 paper, certainly we saw elevated cellularity, but it's possible that that was associated with, forms of direct communication as well. The observation that VEGF-A was dramatically elevated, obviously that's a very tempting conclusion, but it doesn't exclude involvement of other mechanisms. Thank you.
All right, let's go to Dr. Swarup.
Yeah, hi. Yeah, hi, Richard. Very beautiful presentation as always, and a lot of very provocative new ideas. I've heard your white fat stuff earlier, but I didn't read your bioarchive paper, which I just looked at on photoreceptors. Sorry, I missed that.
No worries.
I have a bunch of questions, but one comment first. Being a geneticist, I will take issue with your statement that there's no genetic predisposition to myopia. And just because small effects are there with Jivas doesn't mean it's not there. In fact, most common diseases, which we call multifactorial, are really a combination of genes and environment interaction. So environment, not everybody who goes in certain kind of light will get myopia. Severity probably will change with environment, but predisposition is something different. So I think, what is causal, what is not, I think you can get exposed to all the non-blue light, but if you don't have genetic predisposition, you probably will not. Francis Collins once said that even trauma has a genetic component because response to trauma is highly controlled by genetic variants. So that's something more maybe different. But let me ask you the question that I was sort of thinking about photoreceptors. What you are saying, if that is correct, then we showed earlier that in mammals at least, all rods have a footprint of S cones that was shown by linear staging, but it's not true in case of zebrafish. And a lot of cone genes actually are expressed in early rods. We did extensive studies in single cells and all that. Many of these cone genes are going down, and only by P21 or after, when rods are fully mature, these cone genes are shut off. So if that's the case, is GRIC3, and I'm sorry that I didn't know this paper earlier, I would have checked it myself. GRIC3 in that case should be more in S cones, and I was wondering if in your knockout, maybe, when you said the cones are not altered, maybe did you specifically check as cones and if they have any change in their number?
Yeah, so we have not specifically assessed S cones. So it's an interesting suggestion. Yeah, we were fully aware of the work you've done on the, the precursor status. So and enjoyed that very much. That was part of our thinking about this particular pathway. But let me come back to your first statement. So I don't mind the idea at all that there are gene-dependent susceptibilities. Not at all. I fully accommodate that in our model, but it's extremely difficult to accommodate the idea that it's the only explanation. And so the idea that there's a multifactorial causation argument, I don't mind at all. But presumably you'll also accept the idea that there is an environmental component, given what we know about myopia.
I would never, ever say that any common disease is caused by just genetics or just environment. Never you will hear in my talk that AMD is genetic or diabetic retinopathy or any Parkinson's, Alzheimer's. This is a combination of a threshold genes versus environment. Genes predispose you to a condition. You know, this guy, who was a big smoker, what was his name? Who died at 100 years? He used to smoke cigar all the time. I forget his name. Someone may remember. He never had any kind of lung cancer, but he said smoking is good. I never got anything. That doesn't mean that smoking is really good. So that's all I will say. It's gene environment.
So I think actually, Anand, we agree then.
Yes, we do.
Smoking is good. So let's go to Kim. There was a movie. Thank you for smoking. Kimberly, are you there? If she's not there, then go to Grażyna and Arun.
Okay, fascinating talk. Thank you. My question is, in your experiments with mice, when you were shining the blue-violet light at them, did you see any changes, a little bit following Dr. Swarup, did you see any changes in the outer retina?
You know, we haven't assessed that carefully. You know, these are studies that are ongoing, and you know, we have to look much more closely at, you know, the molecular events that occur. So we haven't really assessed that yet.
Hello, thank you for your wonderful talk. I learned a lot today. So I am very curious about the retinal progenitor cell death. So in your OPN for null mice, you saw that retinal progenitor cell death was increased. So have you ever looked into the myopia development in those cell models? And on the other hand, in the myopia-induced mice models, have you seen any changes in the retinal progenitor cell death rates?
These are great questions. So first of all, you see in the open for null, you actually see a reduced level of cell death, and that results in the elevated rod complement. But the question of whether the open for null myopia phenotype is a result of the elevated number of rods is a great question. That's what you asked in the second component of your question. So we haven't done that yet, but we've been speaking to Michelle Pardieu about that because, of course, Michelle initially analyzed the opium for null. So that's a great analysis to do. What was the third part? Remind me.
I think you answered the question.
Okay.
Thank you so much.
Thank you for the talk. I have a question. It's about for OP5. Did it use 11-cis retinal as a chromorph? It's possible to modulate the chromorph or some small molecules to adjust the OP5 absorbance shift, activation wavelength shift, just slightly shift it from UV to blue light. Maybe it's possible.
Yeah, well, interesting. So the chromophore itself, I think, is going to be very difficult to regulate. So, I mean, we don't have a lot of information about the way opium-5 deals with the chromophore. But, you know, for opium-4, we do, for example, we have a lot of information, and basically it never gives up at chromophore. You have to try really hard to get that out of the molecule. My Japanese colleagues have shown one very interesting thing about opium-5, and that is that when it is light stimulated, it actually spits out the chromophore. And so there is this possibility that, you know, cycling chromophore through opium-5 is dynamic. And so the idea that you might be able to manipulate the chromophore that's available to opium-5 as a way of changing the way that pathway behaves, it's an interesting one, but we haven't really looked at that very carefully at all. You know, it's worthwhile mentioning, I think, that Michelle Pardieu has shown, so another retinoid, retinoic acid, is clearly involved in myopic progression at some level. Michelle has shown that very nicely. She's also shown that if you just feed a mouse retinoic acid, you can actually reverse the retinoic acid deficiency that comes with some of the mutant mice she's dealing with. So for retinoic acid, it's a real possibility.
Hey, Richard, very interesting work and very thought-provoking. I have many questions, but I'll ask one about light regulation of retinal development. So in rodents, most of the retinal development occurs after birth, but in humans, of course, most of it takes place in utero, and I'm sure you get this question a lot. So can you comment on how much light actually the human embryo is exposed to during development? Especially, I'm thinking of people who would live in cold climates where, you know, we wear clothes all the time.
Right. So certainly you can demonstrate that the human fetus is light sensing by, you know, shining lights and showing that there's a response. That's been done multiple times. There's also epidemiological evidence that there is light sensing in the fetus. And it actually comes from work that I did with the former division director at Cincinnati Children's, Michael Young, who was very interested in retinopathy prematurity. So we published a study showing that if you look at day length as a surrogate for light exposure, you can show that there is an association between the risk of retinopathy that requires treatment and day length. So it's another one of the many diseases in which you have a season of birth dependent risk. Now, coupled with that, it's also very clear that OPN4 is expressed very early in human eye development, and I didn't show the data on the archive paper that is on the rod photoreceptor development. It has those data in it. But you see a peak of OPN4 expression in the human fetus, in the human fetal retina at just about 100 days. And so that turns out to be almost exactly equivalent to the day of birth in a mouse if you do the comparative molecular developmental time course. And so I find all of that pretty provocative. And so, you know, you can also see in the human retina, GRIC3 is expressed and NRL are expressed with a timing that is equivalent to that you see in mouse. So, you know, my bias is that you are seeing light stimulations that explain this, but, you know, to demonstrate that directly is obviously very difficult.
Thank you for that really interesting talk. So I was kind of curious, are there regional differences in rod lineage cell density within the OPN4 conditional knockout? And does that pertain to kind of areas of those hybrid neurite localizations or potentially, you know, central versus peripheral retina?
Yeah, I mean, there's a little bit of regionality in the distribution of the IPRGCs. And this has just been, so the original OPN4 Cree line that came from Kingway and San Vital has some issues. It can be promiscuous. We made a new one that very precisely reproduces the pattern of OPN4 immunolabeling. So we've just characterized this, and we can show that there is a little bit of regionality in the IPRGC distribution. And I don't think I remember exactly what that is, but in other words, can that translate to
like spatial
different distribution in rod photoreceptors? Yeah, it's a great thought, and we haven't assessed that yet.
All right, we have a question from Kim. Kim, are you there?
Yes, hi, can you hear me?
Yes.
Okay. We know which cone opsin haplotypes are associated with myopia and extreme forms of myopia. So can you say that your work suggests that environmental spectra would need to be personally tunable to take into account these genotypes, these haplotype information to design ambient lighting products and personalized technologies that are optimized for the visual health of every individual observer? Is this a individualization issue for the design of lighting and technologies?
I mean, you know, obviously with that kind of variation, you could argue that you might need to fine tune it for individuals, but I don't look at the lighting solution that way. I look at a lighting solution that quite precisely mimics the sunlight spectral power distribution, so it works for everybody. So yeah, I mean, it's a really interesting question, but I mean, for me, the lighting solution is one in which the spectral power distribution closely mimics sunlight, and so it works for everybody.
Okay, we have a question from Gula.
Oh, really nice work. I have two questions. When you talk about the sunlight and myopia, do you take contribution of vitamin D or some nutrients? And then also when you talk about the, you know, staying indoors, there's also change in the focus, right? And those two factors can also contribute. What do you think about that?
Yeah, so the vitamin D question, you know, we haven't addressed that directly. There has been an examination of whether vitamin D is involved in as a risk factor for myopia, and my recollection is that it isn't. And so that's one of the reasons why, we like this model. So then the next question about being inside and having a different focal position, it is quite possible that that is part of, that is a myopogenic stimulus. Maybe that's there's been this argument that near work matters. And I've seen studies that support it and studies against it. So I'm not sure exactly where we are with that. There's one particular study out of Spain that compared the myopic progression of two groups of students, environmental science students who are outside all day and lawyers who are inside all day. And it showed pretty clearly that they obviously had all the same amount of computer time. But it wasn't the computer time that mattered. It was whether they were inside or outside. So that focal position, I think that's still equivocal. And and then the violet light response we see, it's unrelated to focal plane, right? Because you still have that minus diopter lens in front of a mouse eye or true eye, you still get protection. So I think that's probably not a key issue.
So you ended up talking about the non-ocular effects of light. And when you turn around to the retina, though, are those truly retina-specific diseases, or can they be treated systemically?
Right. So, it's a great question. Obviously, a lot more work is required to answer that. But we look upon all of these diseases I'm mentioning to you as one consequence of our lighting environment. And so we think that the solution is going to be one set of improvements in the lighting environment. So that's the approach we'll take.
Hi, Dr. Lang. Great talk. I was wondering what your thoughts are on how important it is, the timing of the delivery of the violet light. My impression from reading things is that if you would do that maybe in the evening, it's definitely not good for your sleep, but it might also be kind of counterproductive with promoting myopia. So I was wondering if you had actually done any experiments in the tree shrew or in the mice where it was changed in the timing of the delivery.
Yeah, we're doing a set of experiments in the tree shrew to address that question. And the original paper on the mouse addressed that question. And it was very clear that for mice, delivering violet light in the evening was the key thing in suppressing myopia progression. But stepping back a bit, if you take sunlight spectra at dawn and dusk, you find that the ratio of short wavelength to long wavelength changes. And particularly if you do violet to blue, you find there's a pretty dramatic change that there's a peak of violet to blue ratio that occurs just after the beginning of nautical twilight. So our thinking is that these opsin pathways are actually going to decode that information. So we very much like the idea that if we can produce a lighting system that effectively mimics those ratios, that it won't matter which time of day we deliver. And obviously the reciprocal occurs at dusk. So I actually suspect that the reciprocal occurring at dusk is going to be an important stimulus for effective sleep. But those are some of my thoughts on this, but we have to address that much more carefully.
Yeah, I really enjoyed your talk as well. My question kind of piggybacks off what Zach was asking. In humans, you get this very bizarre staphylomatous formation in myopia progression. And is there anything known about the distribution in humans and the formation of a posterior staphyloma versus total globe expansion?
I don't even know what that is.
It's basically like an aneurysm of the back of the eye.
Oh,
basically
okay.
Just one part of the eye pouches out.
Okay. No, I mean, I have no information that's useful in answering this question.
Okay.
Sorry.
Then I'll piggyback one last question. The dorsal root ganglion and the trigeminal ganglion are the homes for herpes viruses.
Yeah.
And is there stimulation of these opsins that regulate the immune regulation within those populations?
Yeah.
Because clinically, it doesn't sound entirely clear if UV light does trigger.
Yeah, I don't know whether we expect, any kind of change that would influence herpes virus reactivation, but it's very clear at this point that there are immune cells that express opsin 3. There's been no demonstration that they're actually light sensing, but there are also populations that express OPN5. And OPN5, every time we look, it is clearly an opsin. It's functioning as a photopigment. So the immune questions, I think, are extremely interesting, and no one's addressing them yet.
Oh, Dr. Lang, great talk. I have a common question. It appears to me that looking at the incidence of myopia in children and the importance of UV light, short wavelength light in development of proper development of the eye, it seems counterproductive to recommend to children wearing sunglasses for the time being of the eye development. Can you comment on that?
Yeah, absolutely. So, I mean, there are a lot of things we do with our glasses that are probably counterproductive. And so, I feel like somebody who, I've been trying to convince Russ Van Gelder, who's kind of America's ophthalmologist, to publish an article that, makes very specific recommendations. But there are two things that we do that are a bad idea. We block violet photons, obviously, as you pointed out. And we also have blue blocking glasses. Right now, I think that's obviously a marketing ploy for the spectacle companies, but I think what happened was that there's real information, really useful information showing that if you have blue blocking glasses during the night when you're using a screen, that's actually beneficial because you're preventing your circadian clock from being disrupted. So that's actually true. But then it became blue blocking all the time, which is a really bad idea because during the day, you have to have a strong circadian stimulus. So, the recommendations are basically if you need glasses, you need 2 pairs. You have blue blockers, you wear those after sunset, and you have completely clear lenses that you use during the day. And obviously you don't stare at the sun.
Okay. We still have Asim trying to get questions. Asim, are you there?
Yes, I'm here. Yeah.
That's you. You have 2. There's Asim and there's Akash.
Yeah, I'm Osim. Could I ask a question?
Yes, please.
Okay, so hi, Dr. Liang, is really nice talk. So my question is, in addition to the blue light, some kind of the I don't know is a researcher, but I know in China some hospital they try to use a red light to treat myopia. So I want to know what is your comment for that?
Yeah, absolutely. So there is this red light therapy that's being used quite extensively, as I understand it. And it's the device they use to treat kids who have a myopia progression it's actually a laser red light. It's a long wavelength laser red. And there's a device they put over their eyes and they get exposed for a limited period of time every day. As I have been told, the laser light is quite intensive, and there are some questions about safety that have actually been, there was an article recently from one of our colleagues about the issue of safety with this level of light, but it clearly works. So the data that's been published shows that you can suppress myopic progression with this treatment. There is no understanding about why it works, and that always makes me a little bit concerned, but it clearly, if you look at the study that's been done, it clearly works. So yeah, absolutely.
All right, it was a great session. Thank you very much, Richard, and let's join me in thank the speaker today.